Hi everyone, this is a lab guide for finding important structures of the digestive system and how to identify them. Most organs of the digestive system are present along the gastrointestinal tract that are organs in the mouth and pharynx, esophagus, stomach, small and large intestines. The accessory digestive organs facilitating the function of this system are major salivary glands and hepatobiliary tree, including liver, gallbladder, and pancreas. The gut wall is organized by the respective tissue layers located out from the lumen. The mucosa, which is divided to epithelial lining, loose connective tissue in the lamina propria, and smooth muscles in muscularis mucosae. The submucosa is mostly dense irregular connective tissue with the presence of major mucus gland and lymphatic follicle. The muscularis propria or muscularis externa is mostly arranged by inner circular muscle and outer longitudinal muscle. And lastly, the serosa or adventitia is fibroelastic tissue connected with peritoneum, except for adventitia. There are two sites of enteric neuronal clusters presented in the gut wall, the submucosal plexus, which is called Meissner plexus. It is controlled by the innervated group of the cells that control the secretory and absorptive unit in mucosa. And the myenteric plexus, which is called Urbach plexus, innervates the function of the muscularis externa. In the esophagus, the things to identify are the four basic layers of the GI tract wall, beginning from the mucosa, which contains the stratified squamous epithelium non-keratinized, appearing in the mucosa layer from the lumen. Next to the epithelium lining is the lamina propria. You should identify the specific layer to locate the area of epithelial lining and separate them from lamina propria and muscularis mucosae. The submucosa may contain the esophageal glands, which easily spot in the upper part of esophagus. Try to blow up the layer of muscularis externa to identify type of the muscle fibers. The type of the muscle fibers determine the part of this organ. The outermost layer of the esophagus is the adventitia. At the connection between lower esophagus and the cardiac region of stomach, there is a transition of epithelium from stratified squamous to simple columnar epithelium. The site of stomach tissue appears as the gastric pit and the gastric glands, which also called cardiac gland by specific location. The muscularis externa of the stomach forms 2-3 layers. The inner oblique muscle, middle circular muscle, and outer longitudinal muscle. The stomach tissue presents the invagination of space from lumen as a gastric pit. The pit is lined by the surface mucus cell connected with the short neck and to the gastric gland. Appearance of the typical gastric glands roughly determine two groups of the cell type. The numerous eosin-stained cells in the upper part of the gastric glands, which are parietal cells, and the numerous hematoxylin-stained cells in the bottom of the gastric gland, which are Schiff cells. The gastroduodenal junction, the gastric pits in the mucosa change to the intestinal villi and the intestinal gland. The presence of the mucus Brunner's gland is abundant in the submucosa of duodenum. Characteristic of small intestine presents with long projections of intestinal villi and intestinal crypts with intestinal glands, appearing by shape as the simple tubular gland. As the intestinal villi rise high from duodenum to jejunum, the columnar absorptive cell or enterocytes are the most numerous type in the intestinal villi. Alongside to the enterocytes, the goblet cells appear in both the intestinal villi and the intestinal gland. If you follow the intestinal crypt to the bottom of the intestinal gland, it is the house of the Paneth cell, determined by eosinophilic vesicle in their apical cytoplasm. Stem cells and enteroendocrine cells are not easy to identify by H&E staining. At the center of each villi, within the longitudinal projection of lamina propria, small lymphatic capillary is located. This is called lacteal. Three parts of the small intestine can be compared. The height of the intestinal villi from duodenum reaches the maximum in jejunum and rather becomes flat in ileum. In the submucosa, the duodenum presents Brunner's gland. Nothing specialized in the jejunum, and connecting lymphatic follicles present in ileum. The lymphatic follicles in ileum, which lie in continuation to each other, it is called Peyer's patch. Increased numbers of the goblet cells are also observed in the terminal ileum. At colon, the characteristic of intestinal villi is absent. Within the crypt and the intestinal gland, there are numerous enterocytes as well as abundance of the goblet cell. Vermiform appendix connects with the cecum. It has the same feature as other parts of the colon. There are multiple lymphatic follicles appearing in lamina propria and submucosa. Rectoanal junction is in the anal canal, demonstrating a transition of epithelium from simple columnar of the rectal glands to stratified squamous epithelium of the anus. Tissue of liver is organized by hepatic lobules. Each lobule consists of a central vein. surrounded by hepatic cords and hepatic sinusoid. There are 5 to 6 portal triads bounded at the periphery of the lobule. Each portal triad consists of tubular structures of the portal, venule, hepatic arteriole, and bile ductal. Principal cell type of liver is hepatocytes that are arranging in the hepatic cord. A wall of the sinusoid consists of endothelial cells, blood cells, and Kupffer cells. Kupffer cell is characterized by amoeboid shape that extends the cytoplasm in every projection, large and indented ovoid nucleus. It is a liver macrophage with roles in the host defense. Gallbladder shows the appearance of high mucosal folds which cross over each other. It is lined by simple columnar cell with microvilli. However, it lacks muscularis mucosae and submucosa. The smooth muscle layers are interspersed. A tissue of pancreas contains the endocrine part as indicated by a group of less stained cells, which is called islets of Langerhans. Majority of the exocrine part is organized to the acini. Each pancreatic acinus consists of the pancreatic acinar cell, characterized by serous enzyme-producing cells, pyramidal shape with round nucleus. Small pancreatic duct, which is called intercalated duct. This duct penetrates each acinus and the central acinar cells. These cells are the extension of penetrating duct cell. The salivary glands are formed in similar characteristics to the exocrine pancreas as the compound tubuloacinar glands. The gland consists of the salivary acini that can be only serous acini or a mixed type of serous and mucus acini. Each salivary acinus connects with salivary duct, ranging from small intercalated duct to striated duct and the large excretory duct. There are a few myoepithelial cells capped the periphery of each acinus. By H&E staining, parotid gland shows complete serous acini and salivary duct. Submandibular gland shows the mixed serous and mucus acini. Serous acinar cells are in higher population. Sublingual gland shows the mixed acini. Mucus acinar cells are dominant. The mixed salivary glands present the appearance of serous demilune. It is characterized by the crescent serous cells located in outermost area of the mixed acinas. Can you identify the duct cells and salivary acina cell? Here's the list of important keywords in the practical session. Please feel free to ask the instructors and do best on your smart job to hunt the histological characteristics of the digestive organs.